All right. So we're going to be going across the hall here and doing this microscopy lab. We will be looking at 4 things in the microscopy lab. We will be looking at a letter E. I know that seems ridiculous. You know what a letter E looks like. But the reason for looking at a letter E is this. We know what a letter E is supposed to look like. Okay? It is something for which we have a frame of reference. That way, when we make the slide of the letter E and put it under the microscope, if the letter E for some reason does not look right, we will know what's happening inside of the microscope. That is, what is the microscope doing to the light as it passes through it? All right? Obviously, we know that a microscope makes things bigger. Okay? What we want to look at today is essentially how. Okay? How does it make it bigger? And we talked about that when we talked about how Hooke's microscope worked. Okay? His first one. We said, really, it was just a physics experiment involving a series of lenses. Okay, so if you have a specimen, and let's say it's just this little oval here, okay, if you put a lens in front of it, okay, you will change the path of the light. So let's imagine that these straight lines here represent the light that's coming, not really coming off of the specimen, but essentially that's what it's doing. Okay, if those just continue to travel in a straight line and meet your eye sees them as a certain size. Okay, but if I can make that light change its path, and as a result, spread out, the image will then appear what? Larger. Because the light coming from this edge, okay, so let's say it goes through a lens here. Okay, so this thing is a lens. Okay, it's going through here, all right, and usually what happens is, as the light gets to this part, it starts doing this. Okay, now this isn't an exact representation of what happens, but you'll learn about that in physics. Okay. All right, so if my eye is up here near the top of the screen now, okay, the light that's come from this specimen has not only changed direction, but is now also farther apart. Okay, now, thinking about this though, the light is kind of crossed in the middle of this lens, and that's okay, because light doesn't destroy itself when it passes through. Okay, but the light that came from this side of the specimen is now where? It's over here, right? Is that going to change how the specimen looks to me looking through the microscope? It is. Okay, so that's something to think about, because one of the analysis questions in your lab is, what happened to the path of the light? Explain why something appears the way it does. All right, does everyone follow me? So this will be kind of important, okay, when you're looking at that when you're doing your lab report. All right. For the problem, okay, how does the microscope allow us to see tiny specimens? You got to rephrase that in your own words, form of a question. Okay, manipulated variable. All right, what are we manipulating here? Yeah, we're looking at different specimens, certainly. I mean, we're not really comparing them in any way, but certainly we're looking at different things. Okay. What about the responding variable? What are we hoping will happen when we look through the microscope? We're hoping things will appear, yeah, hopefully they'll appear different. You tell me how they're going to appear different, okay? But our responding variable has to be something about how our specimens are going to appear, okay? And the controlled variables are everything that remains the same each time you look at something through the microscope, okay? Those you can figure out, all right? Obviously, magnification actually isn't going to be a controlled variable here for this reason. Our first specimen, the letter E, we will only look at on the scanning power lens, because if we blow it up any bigger than that, you can't see all of it anymore. Okay, it's too big, and if you look at it on high power, all you'll see are the little ink blots, okay, of one single part of it. We want to be able to see the entire letter E, so we can see if it's changed in any way other than being bigger. Okay, but other things will remain the same. Probably how you do things would be things to think about. Okay, did you do something the same way every time? What are those things? Okay, stuff like that. For our hypothesis, we want to generate a hypothesis about how a microscope works, focusing on the path of the light. Okay, so in the if part of our hypothesis, we should probably talk about, light, what lenses might do to that light, okay, and then the then part can talk about what is our image going to look like as a result. Okay? Everybody follow me there? So when we go across, you're going to grab a microscope and you're going to carry it with 2 hands. Good. Okay. You're going to get the necessary slides. The slides are actually up on the front counter. Okay, and how many do you need at one time? One. Okay, please only take one at a time because you don't need more than that. And inevitably, when there's a whole bunch of them sitting on the table, someone's looking through the microscope and they push something off because they're not really looking and then it crashes on the floor. Okay, so you really only need one at a time. Come up and get them as you need them. Okay, we're going to make drawings of the letter E. I would say take pictures of them with your phone. That's the best way really to do it. Okay, take a picture of it with your phone. Now that's going to take a little bit of practice this first time. Okay, because it's not as easy as just slapping your phone on the microscope and having the picture show up. You have to do a little maneuvering, okay, in order to get the picture to actually show up on the screen. Okay, then you can take a picture of it, and then you can use that. Okay, either insert that directly into your Google Doc, okay, which I've already put on Google Classroom this morning. Okay. So you can do that. Sorry, I haven't put it on I have to yet. You can just insert it directly in there and then label it. Okay? Or you can draw the whole thing by hand, snap a picture of that, and put it in the lab report. Either way is fine, as long as you have a lab drawing in there. Okay? On Friday, did you guys get to the part about how to do a proper lab drawing? In the pre-lab part? Okay, well, we'll quickly review that after, but okay. The first thing you're going to look at is the letter E. All right, so to make a wet mount of the letter E, and I know you watched this on Friday, but really quickly, cut out a small letter E, okay? One that is lowercase and from the regular print part of an article in the newspaper that's sitting on the front table. Don't take one from a headline, that's too big, okay? We want one that's a regular print, lowercase letter E, okay? You're going to just cut out a single letter E, put it on the slide, use an eyedropper with some water, put some water on there. As soon as you add the water, the letter E will flip over. It's inconvenient, but it happens. Okay, so what you'll do is you can use, I'll have some, probes and tweezers and stuff up there you can use to just kind of get it set the right way on the slide. Okay, it needs to be on the slide, right side up, so that you can read it as it should appear. That way, you have a frame of reference when you look through the microscope, because it will not look right side up and right in the microscope. Okay, everyone follow me there? Right, so you have the letter E now kind of in a drop of water on the microscope slide. You'll put a little plastic cover slip, just a little small plastic square, on there, drop it on, give it a little tap with your finger or a pencil, okay, and the water just kind of squishes to the edge. Okay, and then that microscope slide is done. You have now made a wet mount of the letter E. Okay, why do we bother to put the water on? Why don't we just put a cover slip over the letter E and throw it in the microscope? Okay, if it's a piece of plastic and a piece of glass, and you tilted it all, what will happen? It'll fall off. What can water do? Okay, why? Why can water make it stick? Okay, does it have to do with anything we talked about in the last unit about water being kind of special? Think about that because it's one of the analysis questions. Okay, all right, so I think that should cover it. So you'll do the letter E first, okay, and then you will look at these other specimens, okay. When we're estimating the size, remember we want to estimate about how many fit across. That's something else you're going to have to record, okay. When you're looking at this specimen on, let's say for everything but the letter E, essentially the high power, estimate how big it is and write that down because that's something that goes on your lab drawing, okay. These are spores, that's nothing we're going to look at. Okay, so here's your kind of proper lab drawing, and I saved this one because this is like the best drawing I have ever made. Okay, I know that's sad, but it's the best one I've ever made. Okay, remember that all of your labels should go to one side of the sheet of paper. Okay, they shouldn't be going all over the place because it looks messy. Okay, they should all go to one side and they should all be straight. Okay, if you need to go down and over, you make a straight line, down and over, and then put the label there. Okay, your title oddly goes underneath. The magnification you looked at underneath, okay, and the approximate size underneath that. Okay, your name and the date go in the top right-hand corner of your diagram. Okay, we talked about that already. Okay, so your wet mount of the letter E needs to look like this. All right, so you'll have this piece of rectangular glass, a slide. Okay, you'll put the letter E on there. You'll put your drop of water, that's the blue circle, and you'll just you'll lay the cover slip kind of like this at an angle and then let it go and it'll just fall down onto the letter E and it'll squish the water and then it'll stick on. Okay. All right, these are the three things you're going to be looking at other than the letter E. Okay, so you look at the letter E first. Then you will look at the amoeba. That's this one. All right, and it'll be kind of this color, purplish pink, okay, on the slides that we have. They don't all have this exact shape, and amoebas are all kind of, they're just a blob, really. Okay, they're going to be a purple blob of varying shapes. Some of them look like a starfish. Some of them look less like a starfish and more like this. Their shape is very pliable. Okay, but generally they are a pink blob. You know, there's a nucleus, okay, things like that. That should be the first one you look at because they're the easiest to find. Okay, the second one you look at should be the paramecium. On some of the slides, the paramecium are greenish, okay, but on some of our slides, they're pink and purple. All right, and there's actually a couple of slides where they actually managed to stain them so the different genders showed up as different colors. Okay, so there's brown and purple and pink. I know you're counting that and going, that's too many genders, but okay, paramecium are different. Okay, so yeah, there's that kind of thing there. Okay, and then lastly, you'll look at the Euglena. All right, and the Euglena is the one you want to look at last because it is by far the smallest. They generally have a similar shape to the paramecium, but they're much, much smaller. Okay, the paramecium, if you look really closely on these pictures, you can actually see kind of a fur, okay, around the edges. It's not fur, okay? They're what's called cilia, and they are how it moves. They're little kind of hair-like things. They're not really hair, and they wave them kind of in unison, and that's how they move. Okay, the Euglena is actually a mobile photosynthetic organism. Okay, it's the same shape as the paramecium, but instead of having cilia, it has a flagellum. You know that tail-like thing I said is hardly on any cells? Yeah, it's got one of those right off the end. You probably won't see it because it's really, really small, okay? But it's on there, and that's how it moves around. So look at the Euglena last, just because it is really, really small and be the hardest one to find, okay? You'll want to have had some practice finding bigger things first. All right? Adam, question? I have essentially these pictures posted as the lab data on Google Classroom, but I mean, hopefully I'm not seeing a bunch of lab drawings with my pictures. I'd like to see lab drawings with the pictures you take or the pictures you draw. I'm okay either way. Okay? All right. Yeah, I think that covers all of it. When we get over there, if you're having trouble getting things focused or whatever, just ask for help and I'll come over and give you a hand. I don't want to be focusing all your specimens because I want you to get the hang of doing it, but I can give you some tips and pointers and help and stuff like that. Okay? So we're going to the bio lab. The door is unlocked, so you can just head that way right now. Okay, listen up here, guys. Couple of things to do with the lab report. Okay, if you're going to use the digital pictures that you took, and you probably will want to do that unless you're really good at drawing by hand, okay, you're going to need to email those to your group mates, okay, and you need to email them to their @isidore.redeemer.ab.ca email accounts so they can save them to their Google Drive, and you will of course need to do the same thing. So email it to yourself if you're the one who took the pictures, okay, to your Isidore@Redeemer email account. That way you can save it on your Google Drive, and then when you're doing your lab report, okay, you can insert that picture into the Google Doc, because when you go to insert, it doesn't give you a lot of options for where to get the picture from. All right, so the best place to have it is on your Google Drive, where you can just pull it and put it in there. Everyone follow me on that? Okay, now if you want to draw it all by hand and then snap a picture of it and insert that picture, the same thing applies. Okay, you will have to, okay, take a picture of it with your phone, email it to your Isidore Redeemer account, okay, and then insert that picture into the lab report itself. Is everyone clear on that? Okay, the due date for this lab report is this Friday. All right, you've got lots of time this week to work on that. Okay, we won't have the Chromebooks or anything, but okay, you can come in and schedule help. You can come in at lunch and work on it if you want, or work on it at home, whichever. Just make sure you have it done for Friday. Questions on that? Okay, if you didn't get to look at everything, come and see me in scheduled helper at lunch, and I'll let you into the bio lab to look at them again. All right, now you still have a couple of minutes, so books out, books open, getting something accomplished.